Okay, so we resume with Ed Munro from the University of Chicago. Ed will be talking about dynamic coupling of adhesion, signaling, and force generation during neural tube closure in an organism we'll be introduced to in a minute.
Good.
Thank you.
Thanks, Thomas. Thanks for putting together a really wonderful symposium. I've already enjoyed the first couple of talks and talking to people, seeing old friends. So Ava gave a nice introduction to the actomyosin cytoskeleton, which saves me a little bit of time. So I can dive right into tissue level and to say that, that embryos use actomyosin contractility. Oh, sorry, I have a tendency to pace when I speak. Embryos, I'll pace very slowly. Embryos use actomyosin contractility in a large variety of different ways to drive different modes of tissue deformation. For example, localizing actomyosin or activating actomyosin contractility at the apical surface of the epithelium can drive bending, or planar polarized activation of myosin at tissue at cell contacts can drive polarized cell intercalation and tissue elongation, or activation of myosin contractility at tissue boundaries can drive tissue closure during wound healing or during development. So a key question in all these cases is, how does actomyosin contractility pattern in space and time in embryos? And because the forces that actomyosin contractility produces also leads to changes in cell shape and tissue architecture, which can feed back to regulate actomyosin contractility, we really want to understand how tissue morphogenesis emerges in a robust way through a dynamic interplay between signaling, force generation, and tissue deformation. So we've been addressing that question in the context of a very simple organism, ascidians. Ascidians are members of the chordate phylum, so they're members of your phylum by virtue of the fact that they develop from simple fertilized eggs into swimming tadpole larvae that have all the basic features of the chordate body plan: a notochord, axial muscles, a dorsal nerve tube. They make these structures with very few cells very rapidly. So one of the advantages of this system is that it's possible to understand how a relatively small number of cells, a few tens of cells, can collectively bring about some very complicated stereotype form of morphogenesis. So we've worked on various different aspects of morphogenesis in this system over the years. The one I want to talk to you about today, as a particular example, is neurulation. So ascidians make a neural tube with about 40 cells in about 2 hours. They do so in a way which resembles very much the way that you made a neural tube, except it took you days and days to make one, and you used many tens of thousands of cells to do it. Here it's 40 cells doing pretty much the same thing. They start out as a flat sheet. The flat sheet invaginates, neural folds come together and meet, and they fuse at the midline and zipper up. Okay? So that's what that zippering part looks like. It's that last step, the zippering up of the neural tube, which I'm going to primarily focus on in today's talk. And this is just to remind you, to tell you about the people who did the work. This work was initiated by Francois Raban when he was a postdoc in the lab, together with Kristen Sherrard, and it's been carried on by Hide Hashimoto in my lab. And I'll tell you primarily about the work that Hide has done more recently. Okay, so we can think about this process of zippering and neural tube closure in quite simple terms. At the edge of the neural plate, neural cells make contact with lateral epidermal cells. And as the plate invaginates and quartets of cells come together, they have to undergo a process of junctional exchange in which neural and epidermal cells let go of one another and make new contacts, neural to neural and epidermal to epidermal, which separate the neural tube from the overlying epidermis. And it's that process of local junctional exchange which is propagating across the embryo from posterior to anterior, which we refer to as zippering. Okay? So how is that zippering driven? Well, experiments I won't tell you about told us early on that this was an active process, zippering. And so to begin to look at the nature of that process, we labeled in living embryos the apical junctions of cells. So in these embryos, the way that we express DNA in cells in embryos typically leads to mosaic expression. So you're looking here at an embryo which is expressing a junctional marker on one side of the embryo. Here is the zipper, here's the boundary between the epidermal cells and the neural cells. And when I start this movie, you'll see some clues about the dynamics of the zippering process. So if you follow cells ahead of the zipper, you see that there's a sequence of contractions here, and then here, and then here. There's a wave of sequential contractions, each one of which drives a net translocation of the zipper forward by 1 unit. If you look behind the zipper, the cells are transiently stretched, and then they relax again. We think that relaxation occurs when the cells complete junctional exchange and let go and can move back. So the basic picture that emerges here is one in which you have active contraction ahead of the zipper that's driving it forward, and passive resistance of cells to stretching behind the zipper, which resists. Okay, so what's producing the forces that drive that? Well, an obvious candidate is myosin 2, and indeed we found that myosin 2 is enriched everywhere along the boundary between neural and epidermal cells ahead of the zipper, in particular right where this rapid active contraction is occurring. So myosin is activated in the right place in the right time. Using standard laser ablation experiments, we can map out the estimates of the relative amount of tension on different junctions at different positions with respect to the zipper. And we see that tension is elevated ahead of the zipper at exactly where myosin is enriched as well, suggesting that myosin is producing elevated tension to drive these cell shape changes. We can block myosin itself, or we can inhibit an upstream activator Rho kinase and completely stop zippering dead. So myosin is required to do this. We can go further upstream and look at the GTPase Rho A that activates myosin in many other contexts. And using a probe for active Rho A, we see that Rho A itself is activated right ahead of the zipper, exactly where myosin becomes activated, exactly where the contraction is occurring. And Rho A is then turned off behind the zipper as well. And if we now use tissue-specific expression of a dominant negative inhibitor of Rho A, inhibiting Rho A just on one side of the embryo, only in the cells right along the boundary, is sufficient to completely block zippering, which says that it's local activation of Rho along this boundary driving actomyosin contractility, which is driving this process. So the basic picture that emerged from this is one, as I said before, of active force ahead of the zipper, tissue resistance behind. And we wanted to use simple computational models to ask whether the pattern of tensions that we measure by laser ablation could be sufficient to explain the kinematics of zippering that we observe. And so to do that, we built very simple two-dimensional models in which we model the boundaries of all cells as simple elements that essentially contract actively, representing the actions of myosin, and the resist deformation with an effective viscosity. And we can now essentially paint onto the cell geometry the relative tensions that we measure by laser ablation. We can also put into these simulations the measured frequency of these junction contractions that go in sequence just ahead of the zipper, the mean time for cells to detach behind the zipper. And we can also pin the value of the effective viscosity by tuning that parameter such that the rate of contraction of junctions just ahead of the zipper matches what we measure. So we can essentially constrain every parameter in this simulation and ask, is this sufficient to explain the dynamics? And the answer is that with those inputs to the model reproduces the kinematics of zippering very closely, and also trivially reproduces the effects of blocking myosin activity. But what's nicer is that with a simulation like that in hand, one can also then ask, is the dissipation of resistance behind the zipper also important by simply disallowing the release, the detachment of cells behind the zipper? And what you see when you do that in silico is that now the zipper stalls completely. So you have to detach cells and dissipate the resistance. But in fact, if we examine if we vary the time delay for zipper detachment in simulations and ask, is that a limiting factor? It turns out that here's the measured value. And for a range of values around the measured value, there's no change in the zippering speed, which says that in the embryo, the detachment is tuned in such a way that it's no longer rate limiting for propagation. So what governs the speed of propagation of the zipper is not this resistance. It's detached fast enough. So what emerges from these simulations is a basic view of what drives this unidirectional propagation of zippering. You have very localized sequence of junction contractions, and it's the asymmetric resistance to the force generated by this very local contraction that converts local contraction into unidirectional movement of the zipper. And the key asymmetries are this dissipation of posterior tissue resistance by cell detachment and relaxation, and also the global asymmetry of myosin activity, higher ahead of the zipper and lower behind the zipper. Right? And because a couple of other talks are going to talk a little bit more about tissue viscoelasticity, I want to make one more point that came out of these simulations, and that's that you could ask the question, why do this with this kind of sequential activation of contraction just ahead of the zipper? Why not close the neural tube simply by making a uniform purse string contraction, as occurs in many other systems? So we can just run a direct competition in simulations by comparing the scenario we observe with one where myosin is activated everywhere ahead of the zipper and ask which works better. And the answer is very obvious. This sequential contraction mechanism works far better. And the reason is obvious once you think about it. Here, in this mechanism, each of these individual contractions feeds cells together where they can meet and undergo this process of junctional exchange and then detach and relax. So each unit of contraction sets the stage for the dissipation of the resistance that builds up right behind the zipper. Whereas here, all of the cells are uniformly deforming, and so it takes a much longer to reach the point where any set of cells can detach from the boundary and relax built-up elastic tension within the tissue. So this is a mechanism to locally concentrate where you dissipate tension in such a way as to make the whole process very efficient. Okay? So two questions came out of this initial work. One is, what generates this tissue-level asymmetry in myosin activity such that it's higher ahead of the zipper and low behind? And the other is, what governs this, what appears to be a propagating wave of sequential contractions from posterior to anterior? So we have more answers about this one, and I'll focus on that and say a little bit about this at the very end. So how do you localize myosin activity at this tissue boundary? Well, there's several different ideas about how this might work. The general idea is that there must be some kind of differential expression of signaling molecules on one side versus the other here. So you could imagine, for example, if you put ligands on one side and receptors on the other, that you would localize signaling just to this boundary. So that's one way in which it could work. Another is that you could have homophilic binding proteins, such as adhesion proteins or other transmembrane homophilic binders, such that expressing those binders on one side of the boundary would lead to engagement only between expressing cells and not at this heterotypic boundary. And that could somehow direct activity to this boundary. Okay? So Hide just went through the Sionna genome looking for representatives of these two different classes of signaling molecules, asking which are expressed in the right place at the right time to find candidates. And he identified a member of the classical cadherin family called cadherin 2 in Sionna, which is expressed specifically in the neural primordium before all of this zippering begins. And when he expresses a GFP-tagged form of this cadherin, he sees that it's enriched in exactly the pattern that you would expect between expressing cells, but not at the boundary. So this sets up that this could be potentially a candidate. So how could one now test the idea that differential expression and this homotypic polarized localization could be important for activating myosin and Rho A at this boundary? Well, here we can take advantage of tricks that we can use in the system to drive tissue-specific expression. So for example, this model predicts that if you were to ectopically express cadherin 2 across the boundary so that you equalize expression at this boundary, that you should turn off myosin and Rho here, and you should activate myosin Rho at this new boundary. And that's exactly what Hide sees when he does this experiment. Here we're taking advantage again of mosaic expression to do this overexpression on one side of the embryo, and we can compare it directly to a control side of the same bilaterally symmetric embryo. And what you can see, at the original neural epidermal expression boundary. Here, when we equalize expression, we see a strong reduction in myosin accumulation relative to the control side. Notice also the control side is much straighter, suggesting it's under much more tension than this boundary. In addition, if we look at the ectopically created boundary out here, we see that myosin is activated relative to the control side. Okay? So that confirms both of those predictions. You can do the, and this manipulation completely blocks zippering. Okay? One can also do that same kind of experiment by inhibiting cadherin-2 just in a single row of neural cells along this boundary. So now equalizing low levels of expression here and creating a new boundary there. And under those conditions, I'm not going to show you the results because they're very similar, but under those conditions, we observe the same thing. Myosin Rho are turned off here and activated along this boundary. Okay? So differential expression of Rho is leading to, and this localization, this polarized localization, is somehow leading to activation of myosin and Rho along this boundary. How might that work? Well, one simple idea is that we know Rho is activated by proteins called GEFs and inactivated by proteins called GAPs. A simple idea is that asymmetrically localized cadherin could sequester an inhibitor of Rho A to these contacts and away from this boundary. So again, one can just go through the Sionna genome looking for candidate gaps. And when he did that, he identified a particular gap called gap 2123. It wasn't the 21st one he tried. It was one of the first he tried. But gap 2123 is expressed initially just along this boundary again, starting right before zippering initiates. So it's in the right place. It's expressed in the right place. When he looks at localization of this gap, he sees that it's localized asymmetrically in exactly the same way that cadherin-2 is, suggesting that it could be localized by cadherin-2. Okay? To test that further, Hide went back and manipulated cadherin-2 expression, again, in this row of neural cells, under conditions in which he's also expressing a reporter for gap 2123 to see how its localization changes. And what he finds is that when he removes cadherin-2 just from this row of neural cells along the boundary, now the gap is uniformly enriched at very low levels everywhere along the boundary. If he instead forces high levels of expression across the boundary so that now cadherin is enriched everywhere in the same cells, now he sees uniformly high levels of enrichment of the gap everywhere. So cadherin-2 is locally controlling the enrichment of the gap. And the basic picture that emerges is one where if cadherin-2 is absent, the gap can accumulate at low levels everywhere. If you force cadherin-2 everywhere, now the gap accumulates at high levels everywhere. Under normal conditions, when cadherin-2 is enriched here, it sequesters all of the gap to this boundary and pulls it away from the boundary between neural and epidermal cells. And that's presumably what allows Rho A and myosin to become active. And I apologize because I didn't mean to animate those slides. But finally, Hide can test by knocking out directly the gap itself, removing it directly from these neural cells. And under those conditions, what he now observes is that the polarized activation of Rho and myosin 2 is completely lost. And in fact, it's lost in an interesting way. That what you see is a higher level of accumulation of myosin at boundaries that previously had almost none, and a reduction here, which suggests that the asymmetric distribution of the gap is actually leading to a redistribution of something that's limiting for activating myosin from contacts between neural cells to this heterotypic boundary to drive activation here. And finally, if he inhibits this gap in neural cells, that also completely blocks zippering. So putting all this together, the picture that emerges is one in which you have local differential expression of cadherin in neural cells, which create across this boundary, which leads first to this asymmetric enrichment of cadherin. It accumulates only at contacts where cadherins can bind across cell boundaries. That the cadherin then sequesters a gap inhibitor which redirects Rho A activity to this boundary. And now, if you put this into the context of the whole tissue, you see that this is a way to dynamically couple junctional exchange that occurs at the zipper to a self-adjusting global tissue-level asymmetry in myosin activity. Because ahead of the zipper, this mechanism directs Rho A and myosin activity along this boundary. But behind the zipper, where junctional exchange has occurred, now the neural cells have formed new contacts. And at those new contacts, cadherin 2 recruits the gap and immediately shuts off Rho and myosin activity. So this gives you a mechanism for a global tissue-level coupling of adhesion to contractility that sustains the asymmetry that's necessary to create unidirectional zipper propagation. Okay, so just how much time do I have, Thomas? A few minutes. Okay, that's all I need. So in the last few minutes, I just want to say a few words about what it is that generates this propagating A to P sequence of contractions along the neural epidermal boundary. So the short answer is we don't really understand how this works yet. But we've been able to rule out a number of mechanisms, I'm not going to show you the data, that might suggest themselves to you immediately, like a trivial mechanism that there's some developmentally encoded timing sequence so that cells further anterior are programmed to contract their boundaries later than the others. So we can rule that out by cutting the embryo in half and showing that there's no, that the cells ahead of the zipper, the cells ahead of the cut no longer contract at all if they don't receive a signal that's coming from the posterior. We can also rule out the idea that, to me, was the most interesting one initially, which is the idea that there's some kind of mechanochemical relay in which local contraction of one junction puts the next one under tension that excites that junction to activate Rho and contract itself and so on. So we can rule that out as well. And that leaves the basic idea that there must be some kind of signal which is propagating from cell to cell, from posterior to anterior, which is activating myosin 2. And here I just want to show you sort of what is to me a very simple but really revealing experiment that Hide did. So in this experiment, he just ablates one cell on one side of the embryo ahead of the zipper and observes what happens. And what you'll see is very clear. The zipper propagates up to the point where it meets that cell, and then it stops. And if you look, you see there's no propagation of contractility along this boundary over here, and therefore there's no signal that's propagating along individual boundaries here. This implies that there must be some kind of communication across the midline, such that this cell has to communicate something to that one and vice versa. This is a very strong implication of this result. And this is just a control showing that if you ablate a cell behind the zipper, nothing happens at all. So that's very suggestive. And we have another very suggestive observation, which is that if you look now at cells just ahead of the zipper, and now we're using a probe that a ionoformin that detects localized protrusions, we can see that there's actually localized protrusive activity just ahead of the zipper, in which cells are extending protrusions across the gap and making contact with cells on the other side. And importantly, that signaling has to be quite short range. It's sort of the range, if it's contact dependent, is limited by the lengths of these protrusions. And that gives rise to a very simple general model for how this might work, which is that there's short range bidirectional signaling across the midline, which excites localized contraction along junctions just ahead of the zipper. And that contraction then advances the zipper forward and also brings cells further along, closer together, so that they're now within signaling range and can signal to one another. And so that's the basic idea that emerges from this, which of course we now have to test further, is that there's, again, a tissue-level feedback loop in which local coupling of short-range signaling bidirectional across the midline to initiate contraction leads to an advance of the zipper, which allows the next set of cells to signal and so on. And I've already mentioned the people who did the work, so I'll just stop there. And I'm probably on time for the first time in my life.